Yes, okay. Okay, good morning. So, welcome to this fourth session in this talks in Introduction to Bioinformatics. I'm very happy to have today with us Cédric Notredame from the CRG, the Center for Genomic Regulation. So Cédric studied biology some years ago in Toulouse, in Paul Sabatier. He did a PhD in bioinformatics with Des Higgins, the author of Clustal, that you know well already. This meant working in Heidelberg and Cambridge. Later he did a postdoc in the National Institute of Medical Research, also in the UK. And then he became scientist at the CNRS in Marseille. And later he became a group leader at the Swiss Institute of Bioinformatics in Lausanne before coming to the CRG. His main interest is development of novel algorithms for the comparison of evolutionary related sequences, and he's the leading author of T-Coffee, another popular sequence alignment program. His team is also behind this tool Nextflow that you see right here, which is very popular in the world of bioinformatics. Probably most of you will use it for the final degree project. So today, actually I forgot, he was also former professor in this degree and coordinator. And today he prepared a talk which is entitled Nextflow, Future Challenges of Bioinformatics in the Large Scale Era. So thank you very much, Cédric.
Thanks a lot, Arnaud, and thank you for having me here. It's always nice to to tell a little bit about these things and what is about to come to the young generation, to the new generation. So today I'm going to talk about what I consider to be some of the main challenges ahead of us, what is changing now. And it so happens that, everything is changing all the time. There's no way back. But every once in a while, the changes accelerate a little bit. And right now is one of these times when changes are accelerating a little bit. And it's worth pondering and it's worth thinking a little bit. What is going to change faster and in which direction? And I'm told there's a little bit of a lag here. Maybe this way? Yes. So the first thing you have to realize is that as we speak, right now there is a race going on. And this race is going on between data growth and computational power. And that's something very important to realize. At the end of the day, all the data, the digitalization of biology, because what was this all about? Biology has always been about data gathering, whether it's Darwin going into the Galapagos Island and shooting every monkey around, or whether it are all the sequencing machines working here and there, data is being gathered. And the comparison of this data, the analysis of this data is going to lead to new knowledge. And for instance, in the case of Darwin, they came back with all these stuffed animals and drawings and all this kind of things. And years of analysis and comparison led to the origin of species, which is arguably the birth of modern biology. Well, the same is going on with DNA. And DNA sequencing initially starting as a chemical endeavor. Sanger was actually a chemist. And it was so successful that it immediately got turned into a mechanized process where you had machines doing the sequencing, able to generate so much output that a single person reading gels will not be enough. And that's when computational biology really started, because data had been generated that could be fed into computers. And as we speak, because in fact, if you follow the technology behind sequencing, you will realize that it's very, very similar to the technology behind computers. When it comes to computers, the main limiting factor for the technology is printing. You have to print things that are so tiny and so fine that you can pack as many circuits as you can on a tiny chip. And the same goes with sequencing. When it comes to sequencing, you have to be able to have chips that are as dense as possible. And so there's a race going on, and this race is between data growth and computer power. And as it happens, computer power has been overtaken by data growth, which means that the amount of DNA sequences available is growing faster than the amount of computational power. And that has real important, and things got worse and worse, you know. So if you look at this thing here, this is the cumulative number of human genomes. And you can tell here that every once in a while, this is already an exponential scale. And even in an exponential scale, every once in a while, these things go exponential. What does this mean? It means that the doubling time is actually shrinking every once in a while. There is so much shrinkage that can take place on the doubling time. Otherwise, you enter in a different, environment, in a different universe. But, you know, every once in a while, these things accelerate. And so what does this mean for us? It means that we have a problem. We have Moore. You know the Moore law? The Moore law is something that was established by Moore, who was an engineer in Silicon Graphics, I think, or Texas Instruments, sorry. And he established that every 18 months, the computing capacity will double. That means that if you wait for 18 months, for the same amount of money, you're able to buy a computer about twice more powerful. That's true for the computation. That's true for absolutely, that's the computation, and that's true for the storage. And as it happens, and this gentleman here is Fred Sanger, as it happens, our DNA sequencing capacity is doubling every 7 months. And that means something very simple. It means that all things being equal, after 18 months, you know, let's say it's 9, after 18 months, in order to be able to process all the DNA that has been sequenced, you'll have to spend twice more money on computation. That's what it means. And after 36 months, 4 times more, and on and on. And you know enough about your growth curve that something growing exponentially is not sustainable, okay? And so if it means that every 18 months we're going to have to spend twice more money on computation, it means that very rapidly we will run out of money. So we have to find solutions. And your generation is going to have to find solutions to address the problem of data growing faster than our capacity to analyze the data. Okay, so the first solution, the simple one, if you're very rich, you buy twice more computers every year. Why not? I love that. The solution number 2, which is more interesting, is that you have to do efficient algorithmic scale-up. Meaning, if you don't have enough money, you have to be smart. You know, you can replace more or less, you know, there is some equivalency between smartness and money. And of course, you can be rich and smart, which is even nicer. But okay, so in this context, this is where we introduced Nextflow quite a few years ago. Now I think we started working on Nextflow in 2014. And Nextflow was meant to be a very efficient way to do large-scale analysis in our lab, taking advantage of the cloud. In these days, Amazon had just started. And if you know Amazon, there are different ways of computing on Amazon. The regular instances are quite expensive. They are like normal computers. But you have all these spot instances, things that will die anytime. So you have to do your computation in a slightly smarter way. And so we wrote a language that will allow us to deploy our computation onto all of these things and make sure that the computation will be robust, it will be reproducible, and making sure the computation will cost us as little as possible. Because that's something, that's a parameter you will all have to take into account. Computation costs money these days. And so an important aspect of Nextflow was that it was supporting something called containers. You may have already seen these things. These are like files that contain an operating system, meaning that you can run one computer inside another computer. And I'll explain in a few minutes why this is so important. And that had to do something with a concept that has become increasingly important. It's called the FAIR concept. Findable, accessible, interoperable, reproducible. You know? And this is what you expect of any computation in biology these days. It has to be done in a fair way. And this has a lot of implication, and I'll deal with this in a few minutes. Oops, different direction. Okay, so what is really Nextflow? You are all familiar now with a Unix shell, and you've all seen this strange little sign here, the pipe. And so the pipe is very central to the way we deal with data in biology. It's funny because computer scientists, they don't really like that stuff. They really like their data to be sort of encapsulated and moved around in a database. You know, computer scientists tend to think in databases, while data handling people like us tend to think in one-liners, in flowing the data. And so Nextflow can be described as a generalization of the pipe. You know, in the pipe you have a program that is producing data, typically line by line, and then you have another program that is able to consume every line of data and do something smart on that line of data. And the pipe is something that is going to allow you to flow data through, to flow data from A that produces it to B that consumes it. And Nextflow is just a generalization in the sense that rather than flowing lines, we are going to flow data structures. We are going to flow elements that can be one genome, that can be one sequence, it can be a graph, anything, as long as you have described it the right way. Okay? And so pipe is based on, and Nextflow is based on something called the reactive programming. So reactive programming is like, you know, you see the surfers from time to time in the Barceloneta, and you see, you know, the life of a surfer is very strange because you're in the water and you wait. And you, and you wait. And every once in a while, the right wave arrives and you jump on the wave. And that's exactly how reactive programming works. We have processes, they're hanging around and they wait. And every once in a while, they receive an indication that data is going to be flown in, and they do something. And then they wait again. This is very different from other ways of organizing your computation where you put the data here and you say, the data is here, let's process the data, okay? Which is a slightly different paradigm. So interestingly, when we started Nextflow, there was another project going on which is called SnakeMake. You will probably hear about it. And SnakeMake is based on the Unix makefile. So what is a makefile? Every once in a while, when you install a program, you type make. And that's actually doing a makefile. And so makefile is very central to Unix. In makefile, the program is first going to generate a dependency graph. And once the dependency graph has been generated, the work will be carried out. Typically, the compilation when you use make, or when you use SnakeMake, the processing of the data. The problem of this approach is that the generation of the dependency graph can take even more time than the actual processing of the data. And also it has a problem that in order to do this, you have to be able to have a perfect overview of the work you're going to do. You're only going to start the work when the graph is finished. And so it's interesting that these days, SnakeMake and Nextflow are the main tools used to do sequence analysis, to do genomics analysis. And it's quite interesting, and I find it funny, that they are based on entirely opposite paradigm. The problem of this, the problem of the makefile, it's great. If you do always the same thing, it's very robust, it's very reproducible, it's quite a nice way. But the scale-up is a problem. If suddenly you're going to analyze 10 times more data or 100 times more data, it may work or it may not work. You have no indication. You have no simple way to know whether your graph is to know whether your graph is not going to be too large to handle everything you want to do. While in the Nextflow paradigm, where you flow the data, you don't care. The scalability is given. You know, the amount of resources is simply going to be proportional to the amount of data you have to process. So, in a way, you can summarize this as Nextflow is actually flowing the data. It's headless. It does not know. It does not need to know how much data it is going to have to process. It simply has to be able to process all the units of data presented to it. While makefile starts by preparing. It has to know, before it starts, how much computation it needs to be done. This can be good if you have to know exactly how much computation or computational resources you need, but this can be, as I was saying, a problem if you're dealing with a very large number of tiny files, for instance, where the actual computation, the actual preparation is going to take much more time than the computation itself. Okay? So this is more or less what Nextflow does, and this is why we created Nextflow. And actually, there were very good reasons for us to create Nextflow. And the main one was that we had all of this pipeline and all of these things that were scattered in the lab, and we had to do something with these things. So, and because we didn't know about SnakeMake, actually. At the time, SnakeMake was still very confidential. So we created our own language. But something more interesting happened. And at the time we were working on Nextflow, there were all these papers about reproducibility. And the community, our community, the biologists, had realized that results were not reproducible. Meaning that, you know, the cornerstone of science is reproducibility. If something is not reproducible, it is not scientific. You cannot make it a scientific result. And, you know, I was talking about Darwin before. Do you know why Darwin and all of these expeditions, these scientific expeditions, happened in the 19th century? It's a funny story. They happened in the 19th century because the 19th century is the only time when sailing became scientific, in the sense that you could know exactly where you were on the planet. Before that, you could have a rough idea that you were going east, west, south, but you could not say exactly where you were in a mathematically precise way. And then in the 19th century, they invented, it's an English who invented, I can't remember the name of this guy, the invention of an accurate clock, an accurate watch that could keep time without losing or gaining seconds for up to a month. And this is essential if you want to know where you are east-west. If you do not have a good timekeeping device, you cannot know where you are east-west. And therefore, you cannot do scientific navigation. You can still discover America, and you can still do all of these things, but you're not going to be able to map things precisely enough to see the drift of the continents and all these kind of things. In order to do things scientifically, you have to be able to go back exactly to the same point and to know that this is exactly the same point. And so the same problem pervades through all science, in the sense that you have the problem that you do not know. If something is not reproducible, you cannot consider it to be a legitimate scientific result. And the whole community was shocked to discover that a lot of the computational results that we were taking for granted were actually not reproducible. And that's where Nextflow proved to have an edge and provide the community with an edge, because it helped addressing the problem of irreproducibility, the fact that you cannot reproduce a computational observation. And this is actually, so we managed to publish this in Nature Biotech. I know it's bad style to brag about the impact factor, but you know the journals do not have, the journals are not equally selective. And so the journals with very high impact factors are more selective, means it's harder to publish in these journals. And this was actually one of the first high impact factor paper of our lab. And we got it into Nature. So Nature Biotech has a very high impact factor because they're actually the one who published CRISPR. CRISPR was rejected by Nature, I think, the CRISPR paper, and Nature Biotech published it. And so suddenly their impact factor skyrocketed. And with these guys, we published this paper in Nature Biotech. And this made it in this journal because of the reproducibility, because we had nailed an issue associated with reproducibility. And what did we see? Well, if you take, so this thing here, don't worry about the details, it's not important, but it's an analysis made on 9,341 genes. If you take a set of expression data, and if you run something called Callisto, which is a very popular quantifier, and you try to figure out, you try to count the number of genes that are differentially expressed, well, if you run this on the Amazon Linux, you are going to find 4,974 differentially expressed genes plus 74. And the interesting thing is that if you take exactly the same data set, exactly the same version of Callisto, everything absolutely identical, and you run it on the Mac OS X, which will be a Unix BSD, I think, you get a slightly different result. You get, in total, 64 and 74 genes that only occur on the Amazon Linux or on the Mac OS X. From a biological point of view, this difference is totally neglectable, you know. It's less than 1%, so you don't care. It's usually not going to affect much your general biological conclusion. But from a computational point of view, this is a disaster, because it means that the two computers are not actually giving the same result. Everything you can control for being equal, you are getting different results. And why is that? I don't know. Nobody really knows. But think about it this way. A computer is like one of the most complex machines we've ever made. You have the CPU, but inside the CPU you have the operating system. And every line of code in the operating system is like a moving part in a machine, in a car or whatever. And so you have all these millions, billions of moving parts. And of course there are slight differences. This can be bugs in the rounding library. This can be anything that is low level. And it results in these very, very slight differences. And that's a real issue, because there are plenty of reasons why you want these results to be identical. Let's imagine for a minute that this thing is an exome sequencing, you know. Now these days, and this is going to be scaled up in the years to come, but if you are suspected to have a disease like a cancer, they are going to sequence your genome to do what is called exome sequencing. And on the basis of the mutation they will find in your genome, a treatment will be proposed. Well, if you go under this paradigm, if you go to one hospital and they analyze your data, they may suggest one treatment, while another hospital, analyzing exactly the same data with the same software, will propose a different treatment. And that cannot be. So you have to, I do not know which one is correct. I have no way to know whether this is more correct than this. These are very, very tiny fluctuations. I simply want the result to be the same everywhere. And that's what can be achieved by using the containerization, by running, rather than running Nextflow on one machine or the other, you run Nextflow inside a container in one machine or the other. And because you run it inside the container, you get exactly the same result. And that's the added value of Nextflow that was entirely built from the start around containerization. Okay, so oops, there I am. And as it happens, you can see very well here, but this became very popular rapidly. And even without us knowing, the Sanger Center next to the EBI implemented this thing here. You don't see very much. It was a companion parasite genome annotation paper where they bundled together up to, I think, 35 algorithmic components. And that was one of the most complex pipelines ever produced at the time. And on this pipeline, we were able to show all the same that if you run it with different flavors of Linux, you're going to get, it predicts genes. It's a gene prediction pipeline. And so on Leishmania, you're going to get a slightly different number of genes. And these differences disappear when you run it in a containerized form. So in these days, we were using Docker, which was the original containerization software. But now there are others. There is Unity and quite a few. So, of course, giving bad news is only fun if you have bad news for everybody. And as it turned out, this instability was actually even true for phylogeny. So this is a phylogenetic tree. These are the branch lengths. And you can see that depending on where you run that stuff. And so I have to insist, a lot of this software, a lot of these algorithms are intrinsically unstable because they use simulation, randomization, okay? So for instance, if you do homology modeling, you know, if you fold proteins, very often the algorithm is going to use a random seed and you will get different results all the time. But this is corrected for. Here, everything is run in such a way that it should give exactly the same result. And in fact, if you run it on the same machine, it always gives the same result. But if you run it on different machines, you have this variation. You're going to say, well, you know, these numbers are not very important. They are so far away from the first digit. But, you know, if you think of the new data sets where we are going to have billions of sequences to align and compare, this is going to end up having an impact on the topology. So this is an issue. This is something that has to be taken care of. Okay. And so the answer of Nextflow was containers, so Docker, and then making sure that everything is published in a traceable way. So GitHub is really a central, you know, something we rely on very, very heavily because it makes everything traceable. It makes it possible to follow updates. It makes it possible to make sure that if you rerun that stuff a week later, you do exactly the same thing with the same operating system and all these kind of things. Okay? And so, yes, these are, this is not so interesting, but these are, the most important thing here was the integration of Nextflow with Amazon. This is something that was very important. From the beginning, what we wanted to do was to be able to deploy large-scale computation on Amazon. Why would you want to do this? You want to do this because Amazon is scalable. If you want to do a small computation, you run it on 5 machines. If you have a huge computation to prepare for next week, you are going to run it on 100,000 machines, as many machines as you have money. Okay? If you have any question or anything, you can interrupt me. Yeah? I'll be happy to answer anything that isn't clear or shocking or whatever. Now, something, and it's more technical. In the early days, it may be shocking, but Nextflow was not modular. The pipeline had to be in one chunk. And this is bad because in reality, what you want your pipeline to be reusable inside another pipeline. You want your pipeline to potentially behave as a function. And so that's why we spend, and Paolo Di Tommaso spent quite a lot of time to push DSL2, which is the newest version of Nextflow that makes it possible to have modules. Oops, the other way around, sorry. Up. Here we go. And so, you know, when I gave this talk this summer, I was at Cubic, and there was this lady, I can't remember her name now, but she works in Munich, I think, and she was giving a talk about benchmarks, and she was saying, never believe people's benchmark. Wait for the next paper where somebody else benchmarks their work, you know, when you compare the accuracy of software or whatever. And so I was very happy to see this paper come out, which was not by any of us, not by any of us, by these guys who compared the usability of all of these pipeline languages. Because you have to know, you know, Nextflow is one of them, but there are up to 300 pipeline languages that have been produced this last year. So everybody is writing pipeline languages. And the zip curve is very, very sharp. I think 90% of the stuff that is published using command line, not Galaxy. Galaxy is quite a big chunk, but everything published command line is done using either Nextflow or SnakeMake. So it's really, these two guys are really, and so here you can see that Nextflow has a bit of an edge over SnakeMake. But to be honest, and when I interact with groups that have already ported their pipeline in SnakeMake, I say, yeah, there's no reason to undo everything you've done in SnakeMake and to push it into Nextflow. These things are covering relatively similar niches. But of course, you know, Nextflow works quite nicely, and it's scalable. I will argue about Nextflow being more scalable. Now, a question we wanted to address with Nextflow, and that we knew was going to require a lot of computation, was looking at instability. I have talked about irreproducibility, and so irreproducibility is about not being able to rerun the same analysis twice. Everything is identical, and you cannot rerun your analysis. That's irreproducibility. But instability is just as annoying. Instability will be that a tiny variation in your sample, a tiny variation in your analysis leads to big variations of the output when it should not. That's what I'm calling here instability. And this is one of the first questions we addressed using Nextflow. With Nextflow, this is the first pipeline we did with Nextflow. And so this pipeline was about turning a bunch of sequences into multiple sequence alignments and into phylogenetic trees. And the idea that we wanted to test the shuffling of the input order of the sequences. You have heard about cluster W, and you've already heard about multiple sequence alignments. And you should know that, in theory, this should have no impact. In theory, if you change the input order of your sequences, it should not impact the final alignment. But it does, and this is a well-known observation. And so we wanted to quantify this impact on very large multiple sequence alignments. And we were quite surprised to find that the impact is very significant. On the difficult multiple sequence alignments, the ones that have between 20 and 30% identity, the ones that are difficult to align, we found that up to 50% of the columns will be different when you start shuffling the input order of your sequences. It's a big result. It's an important result because it says, here is your data, everything is here, and if you do a variation, a very, very tiny variation that should have no impact, you are impacting 50% of your readouts. That's massive. And that is something, a problem for which we have no solution. So if you do not have a solution for a problem like this, what do? You have to be pragmatic. You incorporate this instability in your procedure. You say, well, the stability or the lack of stability should be part of the readout. And whenever I do an analysis, I will decide, I will measure how stable it is. And the stability will be a measure of confidence. If it is very unstable, if the portion of the alignment on which I'm relying is very unstable, I should conclude that, well, I should probably not use this thing. Okay? An important question was, does this have an impact on phylogenetic trees? So phylogenetic trees, you know what they are? They are these things. They are telling you that H and I are sister species, J and K are sister species, and here you have a common ancestor between these guys, okay? And if I have a bunch of sequences that I have organized into a phylogenetic tree, I may want to compare my two phylogenetic trees, and I may want to ask how similar these phylogenetic trees are. And to do this, we have something that is called the Robinson fold distance. You consider that each branch splits your data in two sub-data sets, and so you ask how many split are identical or not. And when we did this, we found that so this is a log of the number of sequences, so that's 10 to the power 3, so that's 10,000, 100,000, and so on. We found that the tree stability is actually surprisingly low. The tree stability is just as low as the alignment stability when you shuffle the input order of the sequences. And bear in mind that the input order of the sequences is something totally arbitrary. There is no good or bad way to decide on the input order. It should have no influence on your analysis, but it does. I won't go further into the paper. Oh, I think I have, yeah, we have an explanation for this, but I won't explain it much here. It's just a fact. The fact is that the way we define the phylogenetic trees, which are binary trees, is actually undefined with respect to the left and the right child, and this is what is causing all of this instability. Okay, and so these variations actually, when it comes to computing multiple sequence alignments, these variations have a huge impact on the final multiple sequence alignment, because even with the same tree, so here I'm going to show here, you see here, this is the same tree. Oops. There you go. Here, this is the same tree. This is A with C, B with D, A with C, B with D, A with C, B with D, A with C, B with D. And so the only difference across all of these trees is that A comes first or second, B comes first or second. But things are still aligned one to another in the same way across all of these things. But because of the way dynamic programming, the algorithms that allows you to align two sequences work, this will induce tiny variations, and these tiny variations will have the kind of influences you see here. Now, these are toy examples. They're not very important or interesting. But if on such simple toy example, you get such a drastic effect by just changing the input order of your sequences, something that should have no effect, imagine how much of an impact you will have when you'll be analyzing 1 million sequences or 1 billion sequences. Things could become very unstable. And that's exactly what we wanted to show in this paper, that there is a lot of instability that is untapped, meaning instability that has not been quantified and that can really give you the wrong impression when you are doing an analysis. Most of the papers doing this kind of analysis, they only do one multiple sequence alignment, and they do not take this instability into account. Now, another aspect that is very important is the non-scalability of accuracy. And here as well, it is something that we have spent a lot of time investigating. So what is a non-scalability of accuracy? So let me show you this. So this is something called the progressive alignment algorithm. I think you see that stuff. I used to teach that stuff in the degree. So I used to teach this, I think, in the second year or something like this. And so you'll see it, your first year students, right? Yeah. So you'll see it next year. So the bottom line is very simple. You have a bunch of sequences, and if you compare these sequences, if you make a multiple alignment of these sequences, it is going to give you a list of all the mutations nature was able to try. And these mutations did not interfere with the organism carrying these mutations being alive. So everybody was alive and the mutations were tolerated. Therefore, the multiple sequence alignment is giving you a list of all the mutations that can be tolerated. It's like experiments made for you by nature. It's amazing. Now, the snag is that the computation of a correct multiple sequence alignment, of a mathematically correct multiple sequence alignment, is what computer scientists call NP-hard. You're probably seeing this in your computer science courses. And so the way we do it is that rather than aligning all the sequences together, we start grouping them 2 by 2, and then we align the sequences 2 by 2. Aligning 2 sequences, we know how to do it. It's called the Needleman and Wunsch algorithm, and then we can align groups of sequences, okay? So this is a progressive algorithm. It's progressive because you align the sequences progressively. Everybody has assumed until about 10 years ago that if you had more sequences, the progressive algorithm will become more accurate, which seems to make sense, you know. It's a little bit denser, therefore the mistakes you will make when aligning these guys with this guy, if you have sequences in between, you know, you have all the intermediate, and it's going to get better and better. And in fact, there were even nice results showing on established data sets that if you increase the number of intermediate sequences, you increase a little bit the accuracy. Everybody was happy. Then this paper came out, and this is the Clustral Omega paper by my PhD supervisor, by Desi Gates. And so the paper came out, I think, in 2011, something like this. And so what do you have here? You have something that is very, very problematic. You have the number of sequences that is increasing, and you have the accuracy achieved on this alignment. And I won't go into the details of how you measure the accuracy and all these kind of things. For here, it does not matter. What matters is that you have a peak here. You have a peak at 1,000, and then it decreases. And that there cannot be any worse graph in data analysis, because this graph is actually telling you something very simple. It is telling you that the more money you spend, the worse your alignments are going to get. And that's a real problem, because normally the more money you spend, the more usable and useful data you are getting. And here, this graph is telling us exactly, so you'd like this thing to go up to 100%, you'd be reasonably satisfied if this was staying flat, but going to lower accuracy when you increase the number of sequences is a real problem. And that's what I'm calling the non-scalability of accuracy. It means that it's not only that you have problems computing very large multiple sequence alignments, it is also that when you compute this very large multiple sequence alignment, not only do you spend too much computation, more than you can afford sometimes, but the results are not as usable as if you were doing this on small things. And I have to insist, I did not mention it, but multiple sequence alignment is one of the most widely used modeling methods in biology today. Among the top 10 cited papers in biology, number 10, you know, number 1 is actually the coloring, the staining of proteins on a gel. And number 10, a few years ago, you know, these things change every once in a while, but number 10 was the Clustral W paper, the one that was published before this one, okay? So these are very, very widely used methods in biology. In fact, there is very little you can do in biology that does not require, at one point or another, a multiple sequence alignment. And so, why? It's very difficult, and nobody really knows why, and we can hand wave on this, and we hand waved on this in our following paper, but the truth is that nobody knows exactly why. And I'll show you here, I'm running out of time, so I won't spend much time on this, but we designed an algorithm that uses the progressive algorithm, but it does a progressive algorithm the other way around. Rather than going from top to bottom, it goes from bottom to up and then goes down again. And that's why we call this thing the regressive algorithm, because it goes exactly the other way around than the progressive algorithm, okay? And so why am I showing you this here? I'm showing it for two reasons. I'm showing it because the scalability of accuracy is going to be a challenge for bioinformatics in the years to come. Something that is accurate enough on a small data set is not guaranteed to be similarly accurate on very large data sets. And I'm showing it here because doing the analysis I'm showing here required millions of alignments to be computed. And for this, we needed Nextflow. And so here I'm showing you, this was our main result. The fact that thanks to this new algorithm, we should go this way, right? But we are kind of able to remain flat as compared with the other algorithms that tend to go down. It's cumulative from right to left, and so that's why you have, it becomes a little bit wobbly when you go there, because here this is only powered by 1 point, while this part is powered by many more points. Okay? So, another aspect that is very important is deployability. You have to make sure that you are able to deploy your computation anywhere. And Alfa Fold is a good example. Everybody wants to do Alfa Fold now because Alfa Fold, it's this new algorithm that makes it possible to predict the 3D structure of proteins. And so Alfa Fold actually starts with a multiple sequence alignment step. And I'm always very proud to think that this guy, Martin Steinegger, who actually contributes the MMSEX part of Alfa Fold, was actually working in our lab. He was sitting in our lab physically when he was preparing the MMSEX algorithm. And so Alfa Fold is a very, very complicated pipeline to deploy. It's very, very complex. And we are now working in collaboration with Martin Steinegger on a way to have on a Nextflow pipeline called Protein Fold that will make it possible to deploy the Alfa Fold algorithm in a much easier way and on much larger, at a much larger scale. So that's something on which we are actively working now. Okay? We were able recently to show, and that's a nice result, that if you use Alfa Fold structures, you get alignment that are as good as if you were using real structures. And that's also something that was a useful result, but I won't spend too much time on this one. It's not so interesting. Okay? So there is one point on which I'd like to talk a little bit and maybe spend a bit more time, which is something that we call readability. And so what is readability? And it's very important, and especially for people of your generation. All of us in this room, at one point or another, we will go to the medic, and our data will be analyzed, will be sequenced. I used to say soon all of us will be rich enough to be sequenced. Now I usually tell rooms like yours, soon none of us will be rich enough not to be sequenced. Because as we speak and as that time passes, all the social security system will want to sequence you in order to be able to assess your risk and to be able to follow your health. And if you refuse to be sequenced, they will say, okay, fine, but then you'll have to pay much more to be insured. And this is something, it's just a matter of time before this happens. And so very soon, none of us will be rich enough not to be sequenced. And one thing that is very important to bear in mind is that we won't be sequenced only once. We will be sequenced on a regular basis. Just like, you're young, so probably you don't do this much, but at my age, you do this kind of every year, and you see yourself slowly decaying, with the analytic on your blood and your cholesterol and all these kind of things. And very soon, we will be followed in the same way for the evolution of our transcriptome and for some local evolution of our genome. And so one problem we have is that every once in a while, unfortunately, you'll be sick. And when you get sick, your genome will be analyzed to see to which treatment you will respond best. And there will always be the situation where there is a magic new treatment that costs a lot, and some analysis carried out on your genome is saying that, oh, sorry, you only have a score of 20, and you'd need a score of 21 in order to be eligible for the flashy new expensive treatment. And as a citizen, you'll feel very bitter about this because you'll think, oh, you know, not only are you sick, but you're not eligible for the best possible treatment. And this number, 20 or 21, it will seem to be coming out of a black box. It will look like something where, you know, why am I a 20? Why am I not a 21? As a citizen, the only way you have to accept this is if you trust the pipeline. And the only way you'll have to trust the pipeline is if either you can read it yourself as a scientist, or if people you trust are able to read this pipeline. And they're able to say, yes, this pipeline is valid science. And I insist on this because we are just coming out of the COVID, you know, pandemic. I hope we're coming out of it. And you've seen a lot going on during the COVID pandemic. You've seen the vax deniers. You've seen a lot of fights around scientific evidence. And I think that the only protection science has against all of the fake news and against all of these kind of things is full transparency. I often have this joke that if news were alleles, fake news will be the fittest by far. Fake news are always much more attractive than real news. It's a fact. Because they are touching on some dark corners of our mind. You say, ah, yeah, maybe. And so we have only one protection against fake news. It is full transparency. You know, it's a matter of fact that every time the state lies a little bit, you know, more often than not, they lie for stupid reason, most of the time incompetence. But anything can fit anything they want into a tiny lie by the state. And that's why we have to press for full transparency. The more transparency you will have, the less likely and the hardest it will be for fake news to be pushed into the dark corners of lies or distorted facts. And in this context, I'm arguing here that readable pipelines for biomedical analysis are part of the transparency. The more readable the pipelines will be, the more transparent will be the medical process. And the more transparent the medical process, the more social engagement we will have. You know, the more support we will have from people in the street who will say, yes, we believe that what you do is correct and what you do is helpful. And so it's much more important now than it has never ever been, because the complexity, the conceptual complexity of health in the decade to come has nothing to do with what it was before. You know, in the years to come, in order to explain, you know, today, in order to explain to someone in the street what is a disease, you have to explain that cholesterol is a molecule, and it goes in your artery, and it blocks them, and you die, and that's bad. And that's relatively simple, because a molecule is just a little thing. But now, in order to explain disease in the years to come, you'll have to be able to explain what is a gene. And it's not simple, conceptually speaking. A gene is absolutely not something simple to explain. But it's an essential concept. And if we do not manage to convey these concepts to societies, then society will distrust its scientists. And this is happening in some part of the world. And so I insist that transparency is just as important, and readability is just as important as the quality of our research. So I was very proud when this came out that about 50% of the COVID data analyzed in the UK, and the UK analyzed like 90% of the COVID data during the pandemic, was actually analyzed with a pipeline that were powered by Nextflow. So these were Nextflow powered data. And so that's one of the things where, you know, you think you have a tiny lab and you're doing something that is only useful for multiple sequence alignment, and before you know it, people are actually doing something very, very useful with this. Shit. Famous last word, and I'll be done. Okay, so yeah, that's a physical joke. It's important as well. So Nextflow then grew into something called NF-Core. And NF-Core, you'll cut this on YouTube, right? Oh no, it's trimmed, damn it. I'm already a meme. And so NF-Core is something that has been built on the top of Nextflow. It's a way of using Nextflow. It's a protocol for using Nextflow to make sure that all of the Nextflow pipeline are high quality and highly interoperable with one another. And you have all of these very nice pipelines that are already there. and of course, all of these tools and all of these things that come along. NF-Core has a very, very rapidly growing community. A few weeks ago, Secura Labs, I will mention and talk about them in a few minutes, Secura Lab, who is now who is an official partner of Nextflow, who is maintaining Nextflow now, and is an official partner of NF-Core. Secura Lab ran the Nextflow Summit, where they managed to have 200 people coming from all around the world, 400 attending remotely, and they rented the Akbar Tower in Glorious. So that's how much these things have been growing in so little time. This is not interesting. We help powering a lot of consortiums. So FANG is, for instance, a consortium on farmed animals to make sure that their farming is as energetically efficient as possible through proper use of genetics. And I want to finish on this because this is quite important. So in my lab, when we did Nextflow, 4 of my students decided to incorporate companies around the Nextflow technology. To me, this was something totally new. In the old days, when I was a young student, if you were good, you would stay in academia, and if you were a bit less good, you would move out of academia. This is finished. Now, you know, some people stay in academia, some people go out of academia. They go to the industry, and there really is no difference. And you can actually, it's because of the level of technology. Going back and forth between industry and academia is much more an option, especially in bioinformatics, than it had ever been. And so these were really my best students. And so Maria Schatzu and Pablo Prieto, who is not here, first incorporated a company called LiveBits around Nextflow. And then Paolo Di Tommaso, who is really the guy who wrote most of the code in Nextflow, and Evan Flodden. Evan Flodden is a student of mine. He was a PhD student, and he's the one who figured out this instability I was mentioning, the fact that you were getting different results on different computers. And so these 2 guys incorporated a company called LiveBits, which is close by here. And these numbers are out of date because last week, these 2 guys managed to raise 22 million euros. This was their Series A, you know, a startup. They go by series, and so the first, the 5 millions were seeding money, and then last week they raised 22 million euros. These guys here, LiveBits, this is not up to date. Now they raised about 80 million euros, and so it's difficult to value exactly how much these companies are worth, but if you look at the market, this means that the combined company of these 2 values is close to 400, 500 million euros. And that's for companies that were incorporated out of a technology grown in a relatively tiny lab. You know, I don't have more than 10 people in my lab at any time. So that's the kind of thing that can happen if you really nail a problem that is required by the community. Big things can happen. And it's, I'm honestly amazed by how successful. I knew that these guys will be extremely successful, but that's beyond extremely successful, you know. It's very impressive. And so these are, you know, the question now is, the real interesting question is whether Secura Lab will be, not the first one, because there was a Catalan unicorn. So you know the unicorn of this company is worth more than $1 billion, and, or €1 billion. And so there was a company, I can't remember what they do, but this is not really a scientific or an academic company. So this one, if it was going all the way it seems to be going, will possibly be one of the first Catalan scientific unicorn coming from academia. And, and so what next? So we are now working on automated way of doing benchmark. When you have a pipeline, it's very important to figure out what is the accuracy, what is the quality of each component. And because these pipelines are increasingly complex, you have to make sure that if you change one component here, you are not breaking the entire pipeline. And that's one of the things we are working on. I'm also chief editor of Now Genomics and Bioinformatics, which is a sister title of Nucleic Acid Research, quite a very well-established journal in biology. And we are just launching a section that will be dedicated to pipelines and that will be dedicated to reproducibility. The hallmark of this journal, the hallmark of Now Genomics and Bioinformatics, is really reproducibility, computational reproducibility. And I think I'm finishing just in time. So a big thank to everybody who was involved in this, and I'll just point a few people. So that's Evan and Paolo, who did live beats. That's Maria Hatsu and Pablo Prieto, who these guys incorporated Sekera Labs, sorry. And Maria and Pablo incorporated live beats. That's Atanasio and Jose, who are now working on this protein folding prediction, on the incorporation of Alfa Fold with Martin Steinegger. And Olivier is a big guy in phylogeny, Olivier Gascuel, and he's the one who helped us on all this stability. And this guy, Phil Ewell here, is working very actively. He's one of the initial creator of NF-Core, and he's now part of Sekera Lab with Evan and Paolo, and working on making sure that all of these pipelines are produced in a way that will be as useful as possible to the community. Okay? And I'll thank you for your attention, and I'll be happy to take your questions.
Okay, so thank you very much, Cédric. Well, we are already out of time. I almost failed. Also, I try to imitate you.
It's full of booby traps.
Okay, I know you have to rush, so you tell us when you...
No, I have until 2,
okay.
So there's a bit of time.
Okay, so as you know, we will start with questions from people in the priority list, and then we will open the general turn.
Hello. What do you think would be the major problem that Nextflow will face on the future?
Well, it's a difficult question. I would say that Nextflow has been so successful that other similar solutions are going to be brought to the market, because if you're very, very successful, then you immediately have competition doing roughly the same thing. What is the major strength of Nextflow, and what struck me as a very, very good decision taken by Paolo and Evan, is that they kept it very open. So first of all, they kept feeding the community, and so the community has a life of its own. It's not something centralized where Paolo, Evan, myself, Phil will decide what can be done or not. People are encouraged to make it their own. For instance, we had a big tension with our funding institute about keeping Nextflow open source, because they wanted to close the source. You know, we had all this discussion every second week. Why don't you close it? It's valuable. You cannot keep the code open like this. Anybody can run with it. And we fought very hard to keep the code open. And I'm 100% sure that all of the success around Nextflow is due to having kept the code open, because that's the way to go. You build a business model on the added value you can bring on your code. And the added value is a community, for instance. And it's typically someone like Paolo, who has spent so much time growing a community. And then when Phil came with NF-Core, rather than being defensive, Paolo gave all the support he could to NF-Core. And NF-Core grew a life of its own. And this, you know, no matter how smart you are, if you have to be smart for everybody, there's never enough intelligence. You know, you have to engage people to be smart next to you. And this way, intelligence gets distributed, and that's how big things happen. And that's exactly the way it went with NF-Core and Nextflow. And so the big challenge to come will be competition. The big challenge to come will be to keep this dynamic going. But, you know, that's something that the future will tell.
Why is the data result so different when we work with...
Sorry, why is what?
Why is the data result so different when we work with different operative systems, and with Nextflow it's always the same? So,
why it's always the same, I can answer. It's easy. It's because these containers, they are like virtual computers, which means that you're actually running the same computer on all your physical computers, you know, because it's in a file. Why the native Unix gives slightly different results, it's difficult, and I do not have an exact answer. I only have speculations. And as I was telling you, you know, if you go into your operating system, you will find that it's full of libraries. It's full of very low-level Unix libraries. And just tiny variation in the version of the library mean that typically, so this is typically the rounding of the float number. You know, you can go to Wikipedia and you will see rounding float numbers is a very, very complex computer science problem. And if I had to guess, I would say that this is the main source of instability we have. And the software is often, you know, in theory, you could write your software so that it becomes insensitive to all these problems. But biologists are not computer scientists, and they do not necessarily write their software to the kind of standards that will be required for their software to be immune to the kind of variations we are talking about here. So it's a combination of practice that are not as good as they should be, and some of fluctuation that are hard to control for.
How long would you say the process was to make Nextflow? And
quite a lot. So I have a blog entry where I traced back all the numbers. But if I remember well, I can't remember now. I think the first attempt to the first version of Nextflow, I think it came in 2013. And the publication of Nextflow, the paper, the Nature Biotech, was published in 2017. And this was not the end of it. Then a lot of work took place to support NF Core, and a lot of work took place for DSL2. So you're talking about easily 9 years of work, and something that is still growing. And I will say, and it's an important question, now we have administrative pressure for all projects to last 4 years. But I cannot think of any scientific project in my lab that lasted less than 6 or 7 years. And that's something that simply means that as a lab, as a PI, as a principal investigator, you have to be agile, and you have to make sure that your project can keep growing despite the turnover of the students. And you have to make sure that everybody gets rewarded for their contribution, whether they are still in the lab or not. But good projects take a long time, much longer than we are told, actually.
Hey, my question, well, you earlier talked about the specific hour watch.
The specific?
Over, like, hour watch.
Ah, the clock, yeah, the watch, yeah.
And my question is, how did they manage to not lose seconds or minutes for a whole month?
Actually, it's a funny story, because I was in, so this guy is buried in Hampstead Heath in London. So if you ever go to London in the Hampstead Heath neighborhood, there is, next to the church, the grave of this guy on whom they wrote the story of his life. And it's a pity. I have the picture. I'll send it to Arnaud. And so I don't know the detail, but I think it's about, it's some piece in the watch that compensates by having a mixture of two metals. And so there is some trick to try to avoid losing seconds. And anyway, the funny thing is that the guy was not a professional watchmaker. He was doing this as a hobby. And so there was a price of £1 million at the time. And so the big people fought very hard not to give him the money. And I think he died. So then there was a suit. And his widow only got a tiny bit of the money. But he is credited for having solved this problem. And so it's, I'll say it's all written on the tombstone of this guy. It's quite a nice story.
Is there really no way to fix the problem of instability? Could it be a line of research in the future?
You know, so if I knew that one of the solutions is scientifically better than the other, then it will be a worthy theme of research, because you know there is a good way of doing things, and you know there is a bad way of doing things. But I don't think the instability I was showing, you know, the graph, the Venn diagram, where you have some genes differentially expressed on Unix and on Amazon Unix and others. If this was, there is no way to tell which one is best. You can only tell that because of low-level variation between these things, you have a little bit of fluctuation. And so you should not bother trying to make everything one way or another. You should just bother trying to make it reproducible, making it consistent across platforms. That will be the main objective here.
Any other question? No? If not, let's thank again Cédric for the fantastic talk.
Thank you, guys.